Okay, so this is where I left off in part one, where we were looking at this idea of passive transport. Obviously something passive doesn't take a lot of energy. So but one way we can kind of think about it is that when you've got a lot of something on one side, it does have energy indirectly because it's energy of position. So a lot of things concentrated are going to tend to move to some area where they are not so concentrated. Water, however, will do the same. However, one way to look at it is it likes to move toward an area that has a lot of dissolved stuff in it, or solutes. When we're looking at solutions that have a membrane in between, we can use these terms to try to understand them. So a solution that is hypertonic means it has a really high amount of things dissolved in it, a high solute concentration. Isotonic solutions mean that basically it's sort of even, it's in somewhat of an equilibrium across the membrane on both sides. And hypotonic means that some solution is very low in solute concentration compared to the thing on the other side of the membrane. This picture is great because it really kind of shows a lot of what we're saying here. But now always think of it in reference to cells. So an animal cell, so the middle frame right here is representing normal cells. Here a red blood cell, here a plant cell, and they're in a solution, or they're surrounded by fluids that are isotonic. In other words, they're pretty much equivalent concentrations of the particles inside the cell versus outside in this area around the cell. And that's true here too. On the left, what you see is an animal cell that's been, a red blood cell again, that's been placed into a solution. So the surrounding solution has lots of particles. This is hypertonic. And because it's hypertonic solution, we're just talking about water now. You might say, well, those particles would want to go in if they could. But one thing we do know is that water is going to go out because water moves in the direction where there's a lot of solutes, high concentration of solute. And the effect on that is what you see here. The cell looks like it's shriveling. Same thing with the plant. Notice that this right here is the cell wall, and here's the cell membrane inside here. So the cell membrane is actually shrinking away from its cell wall for plants. Okay, let's flip it to the other side and see here's a cell, a red blood cell, that's been put into a solution that has high water concentration out here and almost no solutes, very low solutes. And so water is going to tend to want to go in to the cell, which actually will have more solutes than the outside. And what happens is water flows in. And in the case of an animal cell, this can cause it to burst. In the case of a plant cell, it's actually not so bad because a plant cell has that large internal vacuole, central vacuole, and it has the cell wall. So filling up here increases the pressure on the inside, which is called turgor pressure. And that actually helps to support the plant itself. All these cells are nice and plump, and they're actually putting a little bit of pressure. They're pushing back a bit too against that cell wall, and that gives it some rigidity. Here's the word turgor pressure. It's that internal pressure that's pushing against the cell wall. Now we're going to go into facilitated diffusion. So facilitated diffusion is just like it sounds. Something's helping. Something's facilitating materials coming through the membrane, but just passively down their concentration gradients. So the proteins that are embedded in the membrane that are going all the way through, those transmembrane proteins often have channels in them, making them channel proteins. We're going to see how that works right now. So the channels Sometimes there'll be specific channels for just ions to pass through. Now that means that the channel itself has some kind of a charge component. Some of those R groups maybe have a charge on them. Some, though, are gated, meaning that they have to get opened in response to something. There's something called a ligand-gated channel. A ligand is whatever stimulus is. It's usually a chemical stimulus. And then a voltage-gated channel, meaning that the charge of the membrane, there's actually an electrical charge that causes it to open, or the change in that charge. This picture helps a little bit, though. So looking at the picture, here's the ligand here. It's a stimulus. Outside the cell, extracellular side. And this channel, here's this protein channel that has a place for this ligand to bind. It's going to bind here, and you can see what happens over here. And that binding causes the channel to open. And that allows now for certain ions that wouldn't normally be able to pass through the membrane to come through. So this is called a ligand-gated channel because this, the stimulus or ligand, it's not coming in, but it's opening the channel for these ions to flow through. Water is kind of a really particular case because water sometimes can just go through on its own. It can hitchhike through. It kind of goes through with some of the ions that are passing through. But some cells have what are called aqua, water, porins, which are channels that are specific for water to get a lot of water in and out. And you might imagine that an area that might have a lot of aquaporins would be something like your kidneys that needs to process a lot of liquid and water fairly rapidly. This was an experiment that showed that aquaporins actually, when they're present, actually increase the membrane's permeability to water. So permeability, the membrane becomes more permeable, allows more water to go through. And they've created this aquaporin in the membrane. And then they can see what happens if the membrane is allowing more water to come through. Notice it said in a hypotonic solution. That means that the solution that's outside the cell is high concentration of water, low concentration of any solutes. This experiment is in your book if you want to take a look at it. It explains a little bit more on it, but you can see that when they added those channels and they used mRNA to do it. So they had a copy of a little piece of the DNA, that's our mRNA that created the protein, along with ribosomes, and then embedded those into the membrane, and this is what the result was. There are also passive transport by things called carrier proteins. Now, a really important carrier protein are the glucose transporters. And glucose, our cells, they want glucose. They want it. And they will do a lot of things to get glucose to come into the cell and to stay there. So glucose will bind to specific carrier proteins, causing the protein to change shape. And then it will actually, it's kind of like, you'll see it here in a sec, I think, a little way to get it right into the cell. Here it is. Glucose comes in. Glucose binds, and notice what happens here. This is closed on this inside of the cell, this intracellular side, but when it binds, what we get is the movement down. This causes this to come in, and then it spits it out on the interior. So this is a carrier protein that is facilitating the diffusion of glucose. Glucose is in high concentration out here, can't go through very easily. Cell wants it, so it has this protein that is embedded, this carrier protein that not only, it's not a channel protein exactly because notice it's closed over here until it's triggered to open. Glucose is quickly broken down in the cell, so So, and we'll get into this in the next chapter. So we'll see, well, because it doesn't make sense if you think, well, all this glucose is going in, and then isn't it going to just turn around and go back out? But we'll look at how the cell actually captures glucose once it gets in. As glucose diffuses in, we start to have a higher and higher concentration of glucose inside the cell. And you see there's a point which that levels off. Looks similar to what we've seen before. Now this next section is going to look at active transport. And I think I'm going to stop here and restart again with this concept 5.3, specifically looking at active transport. There you go.